So you must have heard about DNA in chapter number 4 where there are like biological molecules. Okay, what is DNA made up of? There, the single unit is known as nucleotide. Okay, now in nucleotide, there are three things in one nucleotide. One is deoxyribose sugar, second is phosphate linkage, and it makes the backbone. And the third important thing is the nitrogenous base, that is adenine, thymine, guanine, and cytosine. Have you learned about these bases already? Okay. A pairs with T, C pairs with G. Okay. Okay. So the backbone is basically of the ribose and phosphate, while the bases are present at the sides which connect the two helix. Okay. So this is one strand and there would be another strand. Opposite to A would be T, opposite to T C would be G, opposite to G would be C, and opposite to T would be A. So there would be similar strand over here with ribose sugar and phosphate, and it would be linked with nitrogenous bases, and these would make bond: cytosine, guanine, and adenine and thymine. Okay. Okay, now the next thing is genetic code. What is genetic code? I'm telling you a simple process which is missed by your book. So I'm telling you a very like simple process. Very first thing is called as the copying of DNA. Copy of DNA. This is known as replication. So when cells divide, there would be copy of DNA. The DNA would be copied and each cell will receive the same number of chromosomes. Okay, do you have any idea about the meiosis and mitosis? Okay, so we say the number of chromosomes remain constant in mitosis, but the number reduces to half in. So how do number remain same in mitosis? Because this DNA replicates. DNA make a copy of itself. Okay, got my point? So the copying of DNA is known as replication. Okay. Then next thing, this is step number 1. Step number 2 is transcription, which is not given by your book. This one is conversion of DNA into RNA, and this is very important process for making protein, because protein is made from RNA, not DNA. Protein is made from RNA. So DNA has to be converted into RNA. Okay. Once it is converted into RNA, this RNA move out of the nucleus. Okay. RNA moves out of the nucleus, and where would it go? To the ribosomes. Where would it go? Towards ribosomes. So you must have heard in chapter number 1 that ribosomes are the protein factories. They synthesize protein. So how do they synthesize protein? When they get RNA. RNA is going to code for protein. Okay. Now RNA get attached to ribosome. Okay. And the sequence of RNA, basically a genetic code, determines the type of protein. Let me explain this in detail. As I've told you earlier that every DNA has genes. A sequence of 3 is known as gene. Okay. So genetic code is also same. A sequence of 3 in RNA is known as genetic code. Each amino acid has a different code. For example, if I say it's CCA, it would code for different amino acid. Let's say glycine. If it's CGA, it would code for a different amino acid, that is alanine. So the sequence will decide the type of amino acid to be put in order. For example, let me write CCA, CGA. Now this is the sequence. Let's say we have TAC and CAT over here. Okay, I'm giving you a random sequence. It's a random sequence. Okay, let's see whether it codes for something or not. Let's say CCA codes for amino acid 1. It's a different type, type 1. This codes for type 5, and this codes for type. How many amino acids are there in your body? 20. 20. 20. Okay, so every part or every gene is coding for a different amino acid. It can happen that they can repeat many times. Maybe CCA can be present 10 times. Okay, then amino acid 1 would be repeated 10 times. Got it? So the sequence of RNA is going, or it codes for it, it is a bit, oh sorry, it is basically a genetic code that gives you a sequence for amino acid. Clear? And these amino acids then bonded together by a peptide bond. Which bond is present between the proteins? Peptide bond. This is in chemistry as well. Peptide linkage, polymers in polymers. Hmm. 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 Okay, it's a part of DNA. Yes, yes. It is in your syllabus. It's a part of your syllabus. Phosphate linkage and ribose sugar makes the backbone of DNA. Okay? There are questions on it, by the way. So yes, they are included. Okay. Then these amino acids join together and make protein. And this process is protein synthesis. This is also part of your syllabus, okay? Okay. I'm just checking what should I teach you next. Okay. Let's move to the inheritance next to it. Okay, till now what we have discussed that there are two type of variation, continuous and discontinuous. Continuous can be affected by environment and genes both, but discontinuous can only be affected by genes. If you change the gene, that would change, otherwise it won't change. Okay. The second thing we have discussed, the DNA. What are the parts of DNA? DNA, how the DNA converts into RNA, and RNA codes for the amino acids or the protein. There is a question, how a protein is made, for 6 mark. So you have to tell about the genetic code, that there is a genetic code, and each code codes for a specific amino acid. Okay. Yes, explain this whole process. And these amino acids are then, okay, first of all, RNA attached to ribosomes, then each this there is a genetic code on ribos or RNA. Each amino acid according to that code would align, and these amino acids would be connected by the peptide bond, and a chain of protein would be formed. Okay. Are you writing it? Okay. Okay, I'm giving you a link, though the structure is not given. The complete detail is not given, but it's like very condensed one. Let me see if I have my own notes. I'll send you that. Okay, for protein production. Okay.